everyone, welcome to the next episode of my science podcast. Today I'm delighted to welcome Lea Goentoro, who is a professor of biology at Caltech, researching systems biology, signalling pathways, robustness and biomaterial. In the lab she studies multiple scales of biological organisation, from small protein circuits, signalling pathways, developmental programs to organisms. So thank you so much for joining us today.
Thank you, Martha, for the invitation to be in this podcast. It's my pleasure.
So to start off with, please may you give us a quick overview of your career?
Yes, well, actually, I started by studying chemical engineering. I studied chemical engineering for my undergraduate work in the University of Wisconsin-Madison. My first research project when I was an undergraduate student was researching how to lyophilize sugar solution.
Okay.
It has nothing to do with what I'm doing now. And then I went to graduate school at Princeton to study chemical engineering. But luckily there was a new professor in the department, Staff Schwartzman, and he offered a project studying the fruit fly Drosophila. So that's how I got started in biological research. I was advised by Staff Schwartzman and Trudy Schubach at Princeton. Then after grad school, I knew that I wanted to continue doing research in biology. So I did postdoc in the systems biology department at Harvard with Mark Kirshner. And then after that, started my own lab at Caltech.
Amazing. And so what brought you into science to start off with?
Right. Well, honestly, I never thought about going to graduate school and really didn't know much about what doing research was all about. It just happens that I took a thermodynamics class in my junior year, I think. In that particular semester, it was a professor named Professor Juan de Pablo who taught the class. You know, the professors rotate teaching the class, and it just happened to be Juan teaching that class. And then at the end of the semester, he invited me to do research in his lab. So I didn't know what the research was, so that's why I said yes. Now I found myself enjoying doing research very much. I would come in the weekends and spend all day in the lab. So after a year, Juan suggested that I apply to graduate school. So I did, and then that's how, that's what brought me into science. And so I would say that what brought me into science and importantly kept me in science is really just having good mentors along the way.
Definitely. And so your earlier work, may you explain to our viewers what fold change detection is and how this benefits certain cellular sensory systems?
Right. So, every cell in our body, in order to function, they have to coordinate functions with other cells. And to do that, they have to be able to communicate, right, with each other. So they do that by continually sensing signals in the environment. Signals are usually secreted proteins. Depending on the signals they sense, then they decide to turn on one gene or to turn off another gene, right?
Yeah.
Okay. So in the postdoc work that I did with Mark Kirshner and Uri Alon at Harvard, we demonstrated that what cells sense is not the level of signal, but changes in the level of signal. And we call this phenomenon fold change detection. So I see a cell sense changes and not level of signal. And this benefits the cells because that by sensing only changes in the signal, that allows them to always adjust to whatever basal level is. So there could be you could be in different conditions, and basal conditions might vary, but that's okay because the cells always adjust to whatever the basal level is and only sense change from that basal level.
Right. And so what kinds of sensory inputs does this scalar symmetry occur in?
Yes. So we first found it in 2 cellular signaling systems. And then since then, other labs have shown that fold change detection also occurs in multiple other cellular signaling systems. So it seems to be prevalent in the way cells send signal. And then it was also shown beautifully, in fact, by Tom Shimishu's lab, to occur in how bacteria sends chemical signals, so in chemotaxis.
Right.
And I think in the past, was it 2 or 3 years ago, it was also shown that fold change detection occurs also in the way social amoeba send signal. Yeah.
That's fascinating. And so my next question is, what is Weber's law and how does this relate to your research?
Weber's law describes how Weber's law describes how our sensory systems detect signal relative to the basal condition. Right. So, for instance, right now, doing this podcast with you, because we're in a quiet background, we can speak softly and you can easily hear me. Yeah. But if we were in a loud rock concert, for instance, then we would have to shout to each other a bit to be heard. Right. So Weber's law was first formulated in the early 19th century, and it's been updated since with more accurate formulations. But the basic idea remains, which is that our sensory systems sense changes in signal rather than the absolute level of signal. Analogous to what we found in cellular signaling systems. So relating the two, this old Weber's law and to what we found much later on, it allows us to propose that from the way we sense signal at organismal level, sense our world, to the way each cell in our body senses its immediate environment, the same principle applies.
Yeah, that's brilliant, because on completely different levels, the same thing's happening.
Yeah.
And so my next question, which is moving on slightly, how can a logarithmic sensor be implemented on a molecular level?
Yes. So this is a nice follow-up question to fold change detection. So fold change detection, proposing fold change detection in cells was not easy. It was surprising, and maybe it was hard to believe, because it's not clear how cells can compute fold changes in protein level. Right? Right. To compute a change in the protein level, a cell must remember the previous level of the protein and then detect the present level of the protein, and then compute the ratio between the present and the previous level of the protein. That's the fold change in the protein level, right? And it was not, when we first published the paper, it was not clear how this computation could be done molecularly. Okay, so the first thing that we did was so now several mechanisms have been proposed, now like 10, 15 years later. Right. So, but the first mechanism that we proposed, and this was work with Mark and Uri along, we proposed that a very common molecular circuit in transcriptional networks, it's called the incoherent feed-forward loop, can accomplish the memory and ratio encoding.
Okay.
So, right. So the cool thing about this finding is that this is a very common circuit in cells. Right? Okay. So incoherent feed-forward loop, what is it? It is a circuit where a signal, the input to the circuit, activates a target, and also at the same time activates an inhibitor of the target. So it's very counterintuitive. Why would why would you activate a target and then at the same time activating the inhibitor of that target to repress that target? Okay. So what we found, we did mathematical modeling, and we found that if the timing of the activation and repression is tuned just right, you can get ratio computation out of this seemingly quirky regulation.
Yeah, because that's different to a lot of things that you might see you don't really see pathways which are contradictory.
Right. Hence the name. That's why it's called incoherent.
Mm-hmm.
Right. Exactly. Yeah. So it's I think what's really cool as a side note is what's really cool about bringing in mathematics into biology is that it enables us to understand more about all the seemingly counterintuitive or not immediately obvious ways cells are doing things, and we lets us to understand surprising insights.
Yeah, changes the way we think of things. And so my next question is, what kinds of diverse biological processes will this role of allosteric proteins acting as logarithmic sensors have an effect on?
Right. So to follow up on the log sensor question, previous question. So there's incoherent feed-forward loop, but then, okay, what about log sensors? Another way that we can get full chain computation is another circuit is by having a log sensor coupled with a negative feedback.
Mm-hmm.
So this is what takes us into log sensor. And then this kind of circuit was already proposed in bacterial chemotaxis. So what is a log sensor? A log sensor is a circuit that produces an output that correlates with the logarithmic of the input.
Mm-hmm.
So it's super hard, at least for me, it's hard to intuit how molecules can do this.
Yeah.
Again, we use the help of mathematics.
Mm.
So a student in my lab, Noah Alsman, showed that if you just take established models that describe how allosteric proteins work, then you get logarithmic computation, right? So that means, well, for us, it was fun to show here's another circuit, here's another molecular implementation of fold change detection. But then allosteric proteins mean this is very, very prevalent in cells because allosteric proteins are found in many biological processes and are also important drug targets. So that's, I guess it suggests to us that logarithmic sensing could be just everywhere.
Yeah, universal.
Mm-hmm.
Definitely. And so, how did you come to suggest that some target genes sense SMAD3 level relative to background as a strategy for coping with cellular noise? And can you tell us a little bit of what cellular noise is before we go into this?
Right. So, good question. So, cellular noise, there are many ways to think about cellular noise. So, I think that at the simplest level, the cells are different from one another, right? Cells, each cell has hundreds of proteins, and concentration of these proteins cannot be precisely the same across all cells, or even the same type of cells, right? So there will be variation from cell to cell.
Yeah.
And the cells have to function well with one another regardless of those variations, right?
Yeah.
So then SMAD3. Okay, so how did we get to, right? How did we get to suggest? So one, like I spoke earlier about how full change detection allows cells to adjust to different baseline.
Yeah.
And right, those baseline could be caused, those differing baselines could be caused by cellular noise as well. So full change detection is a way to cope. Yeah, one way to cope that cells can cope with variations in protein levels. Yeah. So then how did we get at least a glimpse or a hint that SMAD3 could be doing this too? It started again with mathematical analysis. Okay. So we there was already a model of the SMAD3 pathway built by another lab. So it's not built by us, so we can't make it to show what we want to see. So we took a model that was built by another lab, and we spent a summer running simulations with the model and doing analysis. And the result was really striking. With this model that was independently built and has already been verified with multiple experiments, it just came out from the model that the full change in the signal is the robust output of the system.
Okay.
Yeah.
So how does the transforming growth factor B pathway link into this? Because I read in your paper that exposure to ligand then stimulates nuclear localization of the SMAD proteins, which then can regulate the target gene expression. So can you expand a little bit on this for us?
Yeah. So it's TGF-beta, and which is a transforming growth factor beta, or TGF-beta, and SMAD. They're both function in the same pathway. So sometimes we call them TGF-beta SMAD pathway.
Okay.
Right, the same pathway. And TGF-beta is the extracellular signal, is the name of the extracellular ligand or signal that inputs into the pathway. And then SMAD3 is the intercellular transducer of the signal in the system. Yeah. Okay. So most cell signaling systems have the same structure, which is that there is an extracellular signal that's being sent by a receptor at the cell surface. And then the signal itself doesn't go into the cell. So what it does is that it changes in the tail of the receptor, sensed by proteins inside the cells.
Yeah.
Right. And SMAD3 is the one that senses, that transduces signal from the surface triggered by TGF-beta into the nucleus.
Okay, that's fascinating. So my next question is moving on slightly from a molecular level to your work on jellyfish. So the question is, how do moon jellyfish respond to injuries, and how does this differ from other species?
Right. So this is now moving on to work in my lab done by my graduate student, Michael Abrams, and a super fun technician then in the lab, Ty Bassinger. Okay, so we observed that moon jellyfish is a marine creature. So we thought we were just going to see regeneration. But to our surprise, when we halved the moon jelly, they do not regenerate any lost parts, but instead they reorganize existing body parts and quickly regain radial symmetry. So we saw this in moon jelly, and we also saw this in 3 other species of jellyfish. So it could be something, something more prevalent than the moon jelly alone.
Okay. And so this is beneficial to the jellyfish because it's quick, or are there other benefits as well?
Yes, that's right. So I think 2 things at least. So radial symmetry, and radial symmetry is very important in these animals because radial symmetry facilitates propulsion, and so the symmetrical contractions of the jellyfish bell produces fluid flow that facilitates propulsion and also facilitates filter feeding.
Okay.
Right? Exactly. So radial symmetry is really integral to the survival of this animal. Right.
Yeah.
So that's one thing, but also we saw that the jellies that regain symmetry, also regaining symmetry helps them develop further into and mature into a medusa. So that's, yes. So I think we think twofold benefits. One is regaining function quickly, and then facilitating further development.
So what forces is this process driven by?
Yes, that was difficult to find, actually. So first we tested if cell proliferation is involved, and there was no cell proliferation in the process, right? There's no increase in cell proliferation. We could block cell proliferation and the jelly still regained symmetry.
Oh, wow.
And then we thought, okay, so maybe there's cell death somehow, and you get the pooling of the body parts because there's loss of tissue in certain parts. And there was no increase in cell death, and we could block apoptosis, and the jelly still regained radial symmetry.
Okay.
Yes. So the only thing that we found that could block symmetry recovery was when we blocked muscle contraction. Right? So in normal jellyfish, there's this big, really pronounced ring of muscle, and then there's also muscle radially extending into each of out into the bell. And so when we block muscle contraction using menthol, which is a muscle relaxant, symmetry recovery was stopped. And then so we did that for 3 days, and when we took out the jellyfish out of the menthol solution, they resumed regaining symmetry. Right. So that led to another experiment, and led us to propose a model where symmetry recovery is driven by forces from the muscle acting on the viscoelastic tissue. Because it's jellyfish and it's jelly. So the way I like to think about it is if you think of stress ball.
Yeah.
If you ever used one of them. Okay, you take a stress ball and squeeze one side of it, and then it extrudes into the other side. So that's how I like to think about how this works. So if you have symmetrical jellyfish, when the muscle contracts, the forces are all symmetrically balanced. When you have a half jellyfish, then the forces are not balanced, and that moves the tissue around until the forces are rebalanced when symmetry is regained.
That's so interesting. So how long does this symmetrization take the jellyfish?
Right. I think that you pointed out, you already noticed this early on. You said the quick recovery. So it can be really fast. It can be several hours. Some can regain radial symmetry within several hours. So overall, the median is between 1 to 3 days.
Okay.
Yeah. So we think that the animals have evolved this alternative way of responding to injury because of the rate, because it can regain function fast. Regeneration would take longer, we believe. And so radial symmetry being so integral to the function of the animals, and perhaps, yes, this is a way to regain function, and it doesn't matter so much to recover all lost body parts as long as you can become symmetrical again.
Absolutely. Well, thank you so much. That's all my questions answered. So would you like to talk about any of your other projects or expand further on anything we've already covered?
Well, I think that's plenty, and maybe we can do a part 2 next time on other projects.
I would love to.
It's been, it's my pleasure.
Thank you so much. And thank you everyone for listening. I'll see you next time.